welcome everybody to one of the most important grand rounds of the year. This is the Trainee Research Award presentations to celebrate and showcase some of the most important research and some of the best research that's being done in the department. And it spans the gamut. And so for that, I'd like to thank the committee. We also have to have a number of people doing different kinds of research to explain to each other some of what this means. Thank my co-chair, Eric Hung, and then the members of the committee, Andy Crystal, Marina Tulasham, Steve Hinshaw, Dan Mathalon, Dave Manoli, and I hope I didn't forget anybody. The one thing I will ask you to do is to please put your questions in the public chat. And just to remind everybody, it's a very difficult to choose from some great applications, but please keep them coming for next year. Think of it, we particularly want people that are in their final year, priority for people who are senior researchers in their final year, and for work that's being done at UCSF. We'll have David Kastner and his mentor, Lauren Frank, will introduce David.
It's a great pleasure to be here today. Thank you for asking me to do this. David is an extraordinarily impressive guy, and it's really easy to introduce him because I could just read his CV for a while and then we'd be done, but I'll try not to do that. Instead, give you kind of a sense of him. He did his undergraduate degree at Yeshiva University with, I believe, a triple major in biology, chemistry, and English. I don't think that was a single program. Graduated summa cum laude from there and then went to Stanford, where he did his MD, PhD. And the way I sort of think about David's trajectory is as a broad goal of how does the brain do stuff? How does it compute? How does it process information? We all know that the brain is what drives behavior. So in the long run, if you want to understand how behaviors and brain states go wrong, you have to start by understanding brain computation from that perspective. And he did beautiful work with his advisor at Stanford, looking at how the retina, one of sort of the most basic circuits or most fundamental circuits of the brain, computes things about the outside world. He then did a Fulbright scholarship working on computational approaches to learning, and then came to my lab where he's looking at the other end, which is, okay, if we really want to go from computation to behavior and understand maladaptive computation, we also need to understand behavior the same way we understand that computation. And here, David recognized that our understanding of behavior is kind of pathetic, to be perfectly honest. We do things like just say, oh, the animal's doing this, or even the person's doing this, and we know what's going on in their head, so we're going to assign it this sort of label. And that's probably wrong most of the time. You know, if you think about the complexity of the brain, it's not surprising that behavior is equally complex, but our tools for describing and understanding behavior do not measure up to the complexity of that output. And so David has tackled that head on. And I'll say this is probably the work he's done in my lab is almost certainly not stuff that we would have done without him. So he's really pushed us in a new direction. In addition to the work he's going to talk about today, he's done things like work with physicists at Lawrence Berkeley National Lab on new imaging techniques for high resolution imaging of our animals' brains. We can now look at them at 7-micron resolution with their electrodes intact, something we couldn't do before. He's developed new automated behavioral approaches, worked with Peter Dayan, an expert in computation, work I'll talk a little bit about today, about turning behavioral observations into models, and models that can eventually then actually be used to link up to the neural activity and hopefully provide a comprehensive account of how the brain generates behavior and what goes wrong when the behavior isn't what you want it to be, and therefore how you might think about intervening at the neural circuit level if you wanted to fix it. So with that, it's a deep pleasure for me to have David in my lab, and I will turn it over to him to tell you a bit about really actually a small fraction of the work that he's done so far.
Lauren, thank you so much for those kind words. I hope the talk lives up to them. And thank you so much for the committee for this great honor. You know, I've been in the department for a while, and it's just a real wonderful pleasure to get to share some of my work with you. With that, I will share my screen. All right. Just in case there's any confusion, I'm giving a talk with the exact same title tomorrow. It's actually going to be a completely different talk. So if you're so interested, feel free to come. You will hopefully learn new things. So today, as Lauren introduced, I'll be talking about animal behavior and how we try to understand it to leverage it to understand disease. So I have no financial conflicts of interest to disclose. And the start of my work is really behavior. And animal behavior really forms the foundation upon which psychiatric illness is modeled and understood in animals, to the point where it's not at all uncommon. And the paper I'm going to show you is an example, but you can find hundreds, probably even thousands of papers like this, where they have a new model organism. This one is proposing a model organism for the microdeletion of 22q11.2, DeGeorge syndrome previously. And so they have this mouse, they knocked out a gene, and then they want to see what's wrong with the mouse. So they do a whole bunch of different behavioral paradigms: open field, light-dark transition test, prepulse inhibition, spatial alternation, fear conditioning, and they see how these animals differ. And in doing that, they then interpret the features that they find. So in open field, they find that the mice are hyperactive. So that's the interpretation they can provide to understand the phenotype. These light-dark transitions, they are studying the animals being fearful of exploring novel environments. Prepulse inhibition gives us information about this little complicated term, sensory motor gating. Fear conditioning tells us about associative learning and memory. And spatial alternation behavior tells us about working memory. And these are all really fundamental things we want to understand to really understand how this mouse model might relate to schizophrenia or the more complex psychiatric diagnosis that they're trying to model. And the work is fairly comprehensive. The problem is that the interpretation of these behaviors are often quite lacking. And in particular, in this talk, I'm going to focus on two of them: prepulse inhibition and spatial alternation. And I'm going to show you that kind of these more simplistic interpretations really don't reflect the complexity of these behaviors, and ways forward about how to extract more richness from the behavior to better understand these phenomena, and ultimately better understand the way the brain changes in the context of disease to hopefully allow us better understanding of these diseases and ultimately better treatments. So as an outline, I'll first take you through some work I've done about prepulse inhibition, show you that it's more complicated than previously appreciated, show you the extractive features that we now can understand about it. And in the second part, I'll tell you about spatial alternation behavior and how it's intuitively, and the assumption is that it purely reflects memory, that almost definitely is not the case, and what it might reflect so that we can better understand this sophisticated behavior that we have our animals do. So to start, what is prepulse inhibition? So if you hear a loud sound, you will startle, mice will startle, rats will startle. It's a cross-species phenomenon. And if before that loud sound, there's a softer sound, all those species will actually startle less. So it's an inhibition of the startle response due to this prepulse, a preceding sound. And the psychiatric literature has had a lot of excitement in this for, I think, very good reason, in that this differs in schizophrenia. So people with schizophrenia will show less prepulse inhibition. And from the perspective of sensory motor gating, this is a really exciting finding, right? So people with schizophrenia, they hallucinate, they have delusions, they have potentially this disruption in the ability to gate sensation to action. And this prepulse inhibition seems to reflect that. So this prepulse is not processed through as robustly as would be the case in people without schizophrenia. And it gets even more exciting because this phenomenon of PPI actually gets better and improves with treatment for schizophrenia. So with antipsychotics, PPI actually reverts to normal in people with schizophrenia. So here we have this potential biomarker to understand this really complex feature of schizophrenia and maybe even relating to psychosis. And given just how robustly this finding has occurred in schizophrenia, not surprisingly, people have applied it to many other syndromes. Partial list is here. The one I'm going to focus on is Fragile X syndrome, because I've been working with a rat model of Fragile X syndrome where the gene that's responsible for Fragile X syndrome has been knocked out. And so just reviewing the literature of PPI and Fragile X syndrome shows a tremendously inconsistent series of results. So there's a series of papers that show an increase in PPI, the opposite of what happens with schizophrenia. There's a series of papers that show a decrease, the same thing as schizophrenia. And then a small subset of papers, although probably not nearly as robust as with the publication bias would reflect, shows no change in PPI between the Fragile X model mice and wild type mice. And just to highlight just how absurd these inconsistencies are, one of these papers actually studied both humans and the mice and found opposite effects. They found an increase in the mice and a decrease in humans. And the conclusion was, oh, we've modeled the phenomenon in these mice, which was a little hard for me to wrap my head around. So the question becomes, so what are we actually measuring, and can we make these results a little more inconsistent to really try to interpret this, again, robust and really potentially very interesting phenomenon? And to address that, I'm going to be using rats. So as I mentioned, I've been working with FMR1 knockout rats. So FMR1 is the gene that's disrupted in Fragile X syndrome. And we can go and measure PPI. So the way PPI is measured is we put the rats in a tube. The tube has a sensor that's going to measure the movement of the rats. There's a speaker, and we can place different sounds. So we can play loud sounds to which the rat will move and startle, and we can play preceding sounds that will modulate that effect. And the way PPI is traditionally measured is through this ratio, where we take a ratio between the startle that the rat has or any animal has in the presence of the prepulse and the loud sound over the startle just to the baseline, just to the sound itself. And before going on, it's really critical to be able to separate the way we measure the phenomenon, which here is with this ratio, from the phenomenon itself. Of course, the phenomenon exists independent of us measuring it. Our measurements should try to reflect that phenomenon. And what I'm going to show you is that this ratio really doesn't reflect the phenomenon and can be wrong in very problematic ways. So let's think about the phenomenon of PPA, not just the way we measure it. Sorry, just before I do that, let me just show you that I showed you at the beginning, there was inconsistency in the mouse literature. In our hands, we found equal inconsistency in our rats with this PPI ratio. So here we measured PPI across multiple different prepulse levels. And here at 50 decibel startle sound, we found that the FMR1 knockout rats showed increased PPI compared to wild types. In this same cohort of animals, if we just decrease the sound level for the startle, we now find the opposite effect. So now we find that the Fragile X have decreased PPI compared to the wild types. If we do a different cohort at comparable sound levels, here where we saw that there was decreased PPI, now at a relatively similar sound level, we see increased PPI. And at the same sound level where we saw increased PPI before, we now see no effect of the PPI. So the mouse literature is inconsistent. We're able to get inconsistent data with the rats as well. Hooray. But now let's take that step back and let's understand the phenomenon a little bit better. So fundamentally, the phenomenon really starts with this startle response. So here I've just cartooned the startle response as a sigmoid curve. I'll show you that this is actually not a horrible representation. It's actually a rather reasonable one for the rats. And so as the startle sound gets louder, you get more startle, just kind of reflecting what we would expect. So what the PPI ratio assumes is that what's happening to this startle curve is just a scaling at every point. So we can call this startle scaling, and this would be the pure sensory motor gating. And what that does to the startle curve is it scales it down the same amount at each point on the curve. You just take the black curve, divide it by a number, you get the PPI curve. And if this were the case for the phenomenon, the PPI ratio will be perfect, because no matter what startle sound you look at it, you always get the same result, because all that's happening is a division by a fixed number, and by taking the ratio, you can clearly extract out that number. The problem is, if we just think about a sigmoid, division is not the only thing you can do to it. Division on the y-axis is not the only thing you can do to it. You can also manipulate the x-axis. And in particular, this is not a crazy idea, right? We know there's tremendous amount of sensory adaptation, and sensory adaptation would manifest in modifying the x-axis without really doing anything to the y-axis. So if we now add into the phenomenon this idea of sound scaling as well, or modification of the x-axis, where in addition to the division in the y-axis, we also see a division in the x-axis, we now see how the ratio can be very, very wrong. It can give you different results depending on what sound level you get, maybe explaining a little bit of the inconsistent results. So now the question is, can we try to understand if there's startle scaling as well as sound scaling in PPI? And to do that, we need to change the way we measure PPI. Usually people just pick a single startle sound and take the ratio there because you need a ratio, just measure it once. Instead, we now need to measure the full startle curves. So we did that in our rats. And so here's data for the baseline startle curve. So no PPI in a single rat. And as I mentioned, the sigmoid is actually a fairly reasonable description of this phenomenon for just the startle curve itself. We then can measure the full startle curve in the presence of PPI. Again, the sigmoid works really well for capturing the startle of the animal. And we can also measure this across many different pre-pulse conditions. Always the sigmoid fitting quite nicely to the data. And now that we have the data, we can just model it. We can say, okay, let's fit two different models. One model just has startle scaling. One model has both startle scaling and sound scaling, the manipulation of the x-axis and the y-axis. When we do that and we cross-validate the results, so we leave out some data and try to predict that data based on our fit to the rest of the data, we find consistently that the model that has both sound scaling and startle scaling outperforms the model with just startle scaling. So the phenomenon of PPI, not the measurement, the phenomenon of PPI both has a manipulation on the y-axis, the startle scaling that would have previously been thought about as sensory motor gating, but also the phenomenon has within it sensory adaptation, sound scaling that is really critical, and we need to interpret and be able to understand to even interpret what PPI is and why it might differ. So just to show you a little bit more about this in individual animals. So in this animal here, we can look at the amount of startle scaling across different pre-pulse levels. And we see as the pre-pulse level gets louder, you get more of the startle scaling, exactly what you'd expect. The louder that pre-pulse sound is, the less the animal is going to startle to that same startle sound. But the sound scaling also increases such that you're getting effectively more sensory adaptation with the louder that sound, exactly what you'd expect. These trends we can just look at by fitting a line just to see is it going up or down. And consistently across all the animals and conditions, we see the same general feature that with the louder pre-pulse, you're getting more of the startle scaling, you're getting more of the sound scaling. So the phenomenon of PPI is not just this sensory motor gating, it's a mix of the startle scaling, which is what people sensory adaptation. So now by understanding this phenomenon, we can extract out these multiple components and we can go back and ask, okay, is there a difference in these Fragile X model rats? Do we see a difference in the phenomenon of PPI? We now have to look at it in a different way. We can't just look at a ratio. We have much more data. We have multiple dimensions of data. So we have all the parameters of the startle curve, the midpoint, the slope, and the saturation. That defines the sigmoid. And then we also have the startle scaling and the sound scaling, these two features of the phenomenon of PPI. To look at this high dimensional space, I'm just projecting it on all of the marginals. The wild type animals are in the blue color. The FM1 knockouts are in the reddish magenta color. And what you can see is these points overlap completely. There is no difference in the phenomenon of PPI. We can look at that more formally by projecting onto a linear discriminant, which highlights the differences these animals might have due to PPI. And again, absolutely no difference between wild type males and the Fragile X model rats. And this is the case consistently across all of the startle pre-pulse levels we find. We changed the pre-pulse level, so going from loud to softer. We changed the delay, having the pre-pulse sound close to the startle sound itself. And across all of them consistently, we find no difference. In data I'm not going to show, we are able to find differences. So it turns out that the phenomenon of PPI has been shown to differ between males and females. There are sex differences. And with the more complete description, we still can robustly find those differences. So it's not the case that we made things more complicated and we can't see any difference. We made things more complicated and we're likely seeing the real differences and not the inconsistent differences. a critical thing if we really want to utilize these behaviors to interpret what's actually wrong with these animals as we model the human disease. And so that's the first part of the talk. Actually, so now before we move on, so I've shown you that with rats, we have both sound scaling and startle scaling. One of the reasons why PPI is so exciting is because we can also measure it in humans. So thanks to Dan Mathlon and use of his space and just general encouragement to try to adapt our findings to humans, I started collecting data in humans on PPI. So this is the PPI, this is actually my PPI curve. The technician I worked with, Eric Miller, who's the co-first author on the paper, and I are working on this. We have his PPI curve as well. So here we have my scaling to no Prepulse. I happen to startle this amount. It's always fun seeing my data, not just data from an animal that I make do the study. And then this is my Prepulse curve. So my curve decreases quite a bit. And the question is, can we see any evidence for sound scaling here in addition to startle scaling? And it gets a little more complicated. As you see, right, these aren't really sigmoidal. We're not getting up to the saturation level, so it's a little bit harder to pull out these parameters. But what we can look at is we can look at the error landscape of how well a given combination of sound scaling and startle scaling fits these parameters. And what you can see is there's a swath of parameters that equally well fit the data. So this purple line here equally well fits the data with different sound and different startle scaling. It turns out that the best fit has a relatively higher sound scaling and relatively lower startle scaling. But I think that's not the way we want to think about it, right? We should only live within the error of describing the data. And so we can't necessarily definitively say how much, but we can now use these as knobs to turn to try to answer different pieces of information. So the bottom line is that it seems likely that in humans as well, there is sound scaling in addition to startle scaling, gives us the potential to now cross from humans to animals to really understand the phenomenon better and see how it differs in disease. So what I've shown you is that prepulse inhibition of the acoustic star response is more complicated than previously appreciated. It occurs through the combination of both scaling the startle response, the sensory motor gating, as well as the sound, the loudness of the sound. And fundamentally, this PPI ratio is just inherently incomplete and inaccurate. And if we really want to understand the way these things change in disease, we really need to have greater sophistication in extracting the phenomenon. So now on to spatial alternation. So as I showed you initially, spatial alternation behavior is used to study memory. It's used in many different ways. This is one example. So it's a 3-arm track. The animal has to go from arm 2 to the alternating outer arm, so 2 to 1. Then it goes back to arm 2. Since it previously was at arm 1, it has to remember to go to arm 3. Animals can learn to do this task. This is the average of multiple animals. And let's now try to formalize this idea of memory. So without going into too much of the details, we can formalize that with a reinforcement learning model that exclusively has memory in it. When we do that, this model cannot capture the data. It learns too slowly. It can't learn as quickly as the rats. More importantly, it learns in a different way than the rats. So we can split apart the task into 2 components: inbound, which is going from the outer arms, arm 1 and 3, to the center arm 2. And the model makes very different inbound errors than the animals. Or outbound, when it's starting at 2, going to arm 1 or 3. Again, the model makes very, very different errors than the data. This is the best the model can do. The model fundamentally is learning differently than the animals. Memory alone is not describing this phenomenon. So the question is, what is? We can add to the model, in addition to just memory, we can add components of the animal's behavior, such as specific preferences the animal might have. It might like arms more than another. It might like to transition to neighbor arms more than another. When we do that, we now can capture the phenomenon. So it's not just studying memory. This phenomenon of spatial alternation also has features to it, such as spatial preferences, liking certain arms more than another, spatial transitions, liking to go to certain arms more than another. And if you want to understand the variability in the performance of this task, you can't just interpret it as differences in memory. You need to interpret it as other features, higher order features, or different domains of features that are also going wrong or differing between these individual animals. Again, if the idea is to understand how the brain changes, we need to understand the complexity of the behavior to see those differences. I went through that a little quickly, sorry, but to conclude, so I showed you that prepulse inhibition is more complicated than previously appreciated. We have models that can extract these features. Spatial alternation is more complicated. It's not just memory. There are also additional components. And then, of course, to thank all the people involved. The modeling was done with Peter Dion. I've been very fortunate to work in Lauren's lab with a bunch of excellent people. Eric Miller and I did the PPI result. Jojo helped a lot with the modeling, and Anna Gillespie and I collected the data for the spatial alternation. And again, thank you so much to Dan Mathlon for pushing me to do a little human work as well, and all my funding sources.
All right, thank you, David. I was going to break in thanks to a reminder of the time, but you ended with a moment left over. Wonderful. So we have, I guess, 3 minutes, because I gather this needs to end precisely on time, thanks to everyone actually having clinical lives that they need to go to. So if there are any questions, I'm happy to relay them.
Since I can unmute myself, I'm just going to ask David to kind of talk a little bit more about the implication for individual behavior that you've talked about here in the second part.
Yeah, thank you, Susan. And I will be talking about that more tomorrow if you'd like to hear more. But fundamentally, what we're interested in is individual variability, right? That variability could be due to a manipulation we've made, right? Such as a genetic manipulation or something to cause a disease type phenotype, but we want to understand the cause of that variability. It's just as interesting to understand the individual variability within a group of animals, because if we find extremes of that variability, that then could be viewed in some ways as disease itself. And fundamentally, by understanding now that we have at least 3 components of things that underlie spatial alternation behavior, you can think about ways that those can be combined in different ways to give you variability. You can have animals that have better or worse memory. You can have animals that have different types of preferences and utilize them in different ways. And we now have multiple dimensions along which you can get this variability. And of course, multiple dimensions in which it can go wrong and lead to something that might be akin to disease. And of course, by now seeing this complexity, we can start to understand this larger space of variability to really try to understand the ways changes in the brain and the computation of the brain lead to that variability.
Wonderful. All right, we are at 9:29, so I think it's probably my job. Actually, Susan, it's probably your job to say that we need to stop. Is that right?
Yeah, that's thanks. Thank you, everybody. And if you have questions, please contact these trainees directly. And for David, please attend his talk tomorrow at 11 o'clock. Thanks, everybody. And thanks especially to the trainees, and congratulations.